to, again, feel free to ask questions. Let's just not mention any names here. So, we talked a little about last time. Pull the pen up here. Yes, thank you. All right, now the pen should work. I don't know why it makes me go through that, but it makes me go through that every time to get the pen to work on the slides of the smartboard. All right. So this last day of class, again, we've completed all the labs, so no necessarily done with lecture. We're done for the evening. We're done with the semester, so except for test 5. That's what we're here to focus on. Test 5 tonight. We got us about. There's the Hardy-Weinberg principle there in, I believe it's in 19. We didn't get to, and we're just going to put it aside. That's okay. All right, so parts 19 are still valuable. The stuff that we've talked about in class on Monday. We did record that video on Monday, so that's out there on YouTube. If you need help with that, let me know. Okay, but whatever we talked about on Monday, that's the parts of 18 and 19 that I'll cover on test 5. Okay. Let's plan to meet upstairs, 201, the computer lab again, on Canvas. Very similar format. I don't like changing test formats mid-semester. We kind of had to at test 1. Glad that's over, right? So test 5 should look very similar to the other tests, just difference in content, okay? Now, so test 5 will be, again, because there's just not much material there in those 2 chapters, test 5 will be comprehensive. And so it's always worthwhile. What I find when I'm giving a test that is comprehensive, meaning it's going to cover throughout the semester. And when you look at what we call the course outcomes for the course, right? How the course is going to work. What are the expectations for the course? When you look at those, that normally serves as a really nice starting point for a study guide for any kind of a comprehensive test. So if we look at those, number 1, well, it's a common statement there, upon successful completion of the course, the student should be able to, number 1, examine characteristics that are common in all living things and outline the general scheme of inquiry used in science. That's the scientific method, as well as inductive and deductive reasoning, which really I would focus, make sure you understand the scientific method, a little bit about how to apply it, and how it's used. And then be able to, so again, be able to apply scientific inquiry to predict outcomes. And then from there, we moved into kind of a biochemistry section. That was kind of the focus of test 2. We're going to go through all these topics just a little more in depth, but just, again, this is a nice jumping off point of, if you read through these at this point in the semester, and it's like, Steve, I don't know what any of this is talking about, then that means we've got some catching up to do, and that's okay. We just need to identify, pick these out and say, all right, I don't, upon successful completion of the course, students should be able to do these things. Which of these things do not make sense to you at this point, okay? And it's okay if you don't know every single word. We can look up individual words. It's just, in general, concepts don't make sense. That's kind of important. So again, number 3 is identify between chemical components of life. Number 4, analyze differences between various cell types. So understand prokaryotic and eukaryotic cells, plant and animal cells, and then a little bit about cellular reproduction. And then number 5 is describe metabolic processes. That's specifically all metabolism, but especially respiration and photosynthesis. And then number 6, apply principles of energetics to living systems, okay? Which is also, that is the photosynthesis, cellular respiration. And then understand both heredity, the genetics we talked about in test 4, as well as evolution. That's the new material for this test, and how those impact and are passed through organisms, and also how the environment, how the ecosystems also can impact those as well. Okay? So again, that's always whenever we're looking at, and this is just, when I sat down and thought about what's going to be the most helpful for us at this point, And it strikes me as some of you guys are probably going to be very close to pure freshmen. I don't know how many of you have never taken a college course before. So I thought, well, it's probably not the only, it may not be the only comprehensive, well, that's not going to work. This may not be the only comprehensive test you're taking this semester, and it probably won't be the last one you take. So I thought, well, let's just take some time and talk about how would you go about studying for a comprehensive test. That's a good thing to do this evening, in addition to talking about the basic concepts. So it's kind of where we are. Again, if you're taking a comprehensive test, this is a nice place to start. Go into your syllabus and find a good syllabus. Every syllabus, not just a good syllabus. Every syllabus should have these course outcomes. What is it that you should be able to do? And if you read through those, on successful completion of the course, the student should be able to, and something just does not make sense, you as a student, that means you've got to spend some time and see, all right, what the heck is this concept talking about, okay? So it's a nice jumping off point for us. All right. So that's what I'm saying. Let's take this as an opportunity to discuss reviewing for any comprehensive exam. That's going to help you here, and that's going to help you any other tests you're taking that ends up being comprehensive. Where do you start? If you're like me, not everybody is this way. I'm almost to a fault in big picture. I'll zoom out and look at it, try to look at the big picture, and details can easily overwhelm me. You got just enough ADHD that I'll get stuck going down a rabbit hole. It's real easy to get overwhelmed. Maybe that's not you, and that's okay. There's nothing wrong with that. But this is one thing I see this a lot of times in students. When you really take a zoom out and look at the whole picture for a comprehensive test, it's really easy to be overwhelmed. How do you handle that? How do you eat an elephant? Have I heard this old adage? How do you eat an elephant?
One bite at a time.
One bite at a time. One bite at a time. It's really easy to look at something that's massive and say, Steve, there's no way I can do this. How do you do it? Start cutting it up. What are the things you can achieve? What's realistic? What's realistic for you? Every individual is going to have their own ability and time budget that they can deal with for studying. Look at what amount you've got a week. Now you have 7 days between now and when we'll take this test. But not everybody's going to have the same time that they can allocate to this. Sit down, when we get done here, before you leave, sit down and just sketch out roughly what time you think you'll have to study for this test, and then what other tests you have, and plan them. Right? I teach a course in Intro to Engineering where I try to bring in some guest speakers, and more than once I've heard from multiple guest speakers. I'll bring them from the industry, and I've heard this phrase several times. A fail to plan is a plan to fail. Right? I don't know if you've heard that before. You don't sit down and think about when can I actually study for these. Think about that, right? And because when you really look at what you should study, you look at the list of topics we've got, it's pretty easy to just say that's crazy. It's a big elephant. There's no way to eat it. How do you eat it? Carve it up a little bit at a time, and let's attack it. Okay, so what should you study? All right, again, I just took a screenshot from the table of contents of our textbook. Nice starting place. You look at this and say, well, whoa, we've covered 1 through 19. Right? There's more chapters than that in the textbook. We've covered 1 through 19. We've not covered all of it. There's parts of it we set aside. There's a chapter in here we just didn't cover. That's a lot. Again, let's try to not get overwhelmed. Let's take this apart. Down here at the bottom, the last 2 chapters, 18 and 19, that's what we talked about Monday. And again, I felt uncomfortable making an entire test off 2 chapters we covered in a day. Let's not do that. Let's take the big parts of the other chapters and put that on the test too. I've not written the test yet. I'm hoping you're gathering that. But what I'm planning for roughly in my head is about 40% of the test will be from chapters 18 and 19. About 40% of the points. That leaves 60% of the points to be spread out between the other 4. So I'm thinking roughly 15% per other test. That really just leaves room for the big topics. Okay, that's my goal. I'm not putting that in writing because I'm not promising that's exactly how the points will shake out, but that is my goal, and I should be able to get pretty close to that. Right? But no more that I assure you, no more than 50% of the points will come from these two chapters. Right? So it's still significant. You can't blow off these last two chapters. Just again, that's kind of what we're looking at. Okay. So let's look at the other. So prepare for that portion of this test, just like you would for the other tests. And I say just like you would. I would suggest if you're not happy with how you've done on the other tests, then we probably need to try to figure out some other way to prepare for this test. Okay. If you're happy with how you perform on the other tests, then just rinse and repeat, keep up, and you should be just fine with this portion of the test. Okay. As far as the rest of it, how do you study for the rest of it? And again, I probably say this. I think I'm going to say this about 3 times. What we talked about earlier. That's 19 chapters, Steve. That's a lot. Yeah, it is. It is. But you're not starting from scratch. You're starting from experience at this point. You've seen all this other material. You're not having to completely build it up. We just need to figure out how do we break this elephant into smaller pieces so we can get through it. Okay. Good news, we didn't do 17. We're not going to do 17. So cut that one out. So it's just 1 through 16 and then 18 and 19. We didn't do, honestly, if you look at what we covered as opposed to what's contained in 13 through 16, there's a lot of 13 through 16 that we just didn't get to. We went through quickly, and it was a little more in depth than we needed to go. Okay, but we covered some of the high points of all those. Okay, so I'd suggest 3 things as far as this old portion, right? Chapters 1 through 16. Again, the 18 and 19 do just as you've done on the other exams. Read your notes, read the text. I put out assignments. I put out a chapter 18, 19 assignment. It's, as usual, it's very, very high level overall questions. There's a chapter 18, 19 quiz. And how do we handle the rest of it? The other chapters, the other 16 chapters, how do we handle those? Well, old test is a nice place to start. You should be able to my knowledge, you should be able to go into Canvas, look at your old tests, look at how you did, okay? Look at your lecture notes. Some of these videos are recorded. Some of them I've recorded here. Some of them we had to be online, so they're recorded there. I don't know that I would watch those at regular speed. You can go into YouTube and tell it to 1.5 speed or 2 speed. A lot of people, I hear from a lot of people that they can go at about 1.5 to 1.75 speed with the closed captioning on, and they normally do okay. But if that helps you do that, I go slow enough to give you a chance to process the information that reviewing it, I probably wouldn't go back and watch it at regular speed. Okay, but review your old lecture notes, the lecture videos if those are helpful. Make sure you look at, if you haven't already figured out, the homework questions and sometimes those quiz questions are a starting place for me when I write the test. Right? I don't mind telling you that. Not every question in the homework or not every question in the quiz shows up in the test, but a lot of them do, or at least parts of them. And then as you have time, so this is kind of in order of priority of how I would attack trying to study for a comprehensive test. Start with your old test, start with your homeworks, your notes, your quizzes, and then as you have time, go back and reconsult the text. That's assuming you've had the opportunity to go through and read the majority of the text as you've gone through. And I know if you're like me, reading a text is it is a very energy intensive process for me. It's I don't know, maybe some of you guys are reading the text as your predominant way of learning. I went to school with people like that. Hats off to you, I'm jealous. That is not my primary means of learning. I can do it, and I've gotten to where I can do it pretty well because of where you guys are right now in your development is forcing myself to sit down and really go through a text and try to get information out of it. Okay, it's a little different than reading for pleasure, that's for sure. All right, so again, review old tests. If we break those down just a little bit, when you go back and look through your old tests, what makes sense now? Do all the questions there make sense? Because a lot of what we've done throughout this course really has built on a lot of the previous stuff. Not all of it, but a lot of it has built back up. There are some big chunks that we talked about on that test, and then we haven't talked about again, but a lot of it does relate. As you go back through those tests now, do those questions make sense? Do the ones that you got wrong, do you understand what the correct answer is now? Do you understand why, how you can improve that this time, okay? And again, go back through your notes, homework, quizzes, and this brings us up to then the text, right? Reviewing through the text, and that's a lot of material. If you have time to reread all those chapters, I know students that do that, and that's awesome. Again, that may not be for everybody. Maybe A, you don't have the time, or B, you're like, man, it's really rough on you to read that much. If not, whatever the case may be, if you know that you're not going to get through all 16 of those chapters again, that's fine. Let's have a plan. And again, my hope is this is helpful for you in any comprehensive test you're taking. I'm hoping it's probably not the only one you're taking now, and I know it's not going to be between now and the time you graduate. So be systematic with the time you have. You also got to account for what time you're going to need to study for your other tests. This may not be the only class you're taking. Again, it's easy to get overwhelmed. Break into small pieces. Plan to be successful. A fail to plan is a plan to fail. Okay? All right. Break it up into its units. Break it up into the pieces. Cut the elephant into small bites. Look at it in terms of, all right, these chapters went with test 1. These chapters went with test 2, etc., etc. And then try to approach it systematically that way. And if you look and say, well, I did really bad on test 3. Okay? I don't know, maybe you did great on test 3. But whatever test you did really bad on, okay, maybe those are the chapters we focus the most on. Okay? But if you kind of look at it that way, again, it's really easy to zoom out, see everything, and get distracted. Look at it as smaller chunks. These are the chunks you're looking at. Okay? Test 1, 2, 3, 4 chunks, and then the new chunk. So, and this is the next 4 slides here, just repeating the same thing over and over again because it really is rinse and repeat. The test 1 material, that's chapters 1 through 5. At the very least, if you've got time to reread those entire chapters, do it. That's fine. If that's helpful for you, do it. At the very least, read those chapter summaries. They're pretty well written. They're pretty good. They don't cover everything. But if you read the chapter summary and something in that summary just doesn't make sense, take notes. Write it down. You need to learn more about extracellular receptors, or whatever the case may be. And then if you're reading through those summaries and one of those summaries just makes no sense whatsoever, that tells you, all right, that's probably the chapter I need to, if I have time, to go back and focus on. Okay. And again, make sure you've got 18 and 19 pretty well squared away first, and then come back and start working through these other chapters as well. And again, just like I said, this is rinse and repeat. Test 2, chapters 6 through 8. Read the summaries. Take notes. What doesn't make sense? Make note of it. Go back later. I feel like I'm using a lot of old adages today. You guys, I'm sure, have heard the old adage of you can take a horse to water, but you can't make it drink, right? What I'm trying to convey to you is I can't learn this material for you. I can't do it. We've gone through the entire semester, and I've tried to point to the stuff that are important and try to point in the right direction. And kind of hit the point where, again, it's rinse and repeat here. This is kind of where we've got to make sure we've got a good handle on at least the most important things. A lot of times it's the stuff that's going to show up in those chapter summaries. Okay. So again, break this down into smaller pieces. What are the important topics? Okay. And work your way through. Now, it's been a hot minute since we took test 1. We had to take that one online, remember? Buildings were shut down and the world was frozen. I had to let it go. Anyway, test 1. Talking about defining life, defining science, defining biology. What are these? And that's kind of chapter 1. We talked about the criteria to be alive, what life actually is. And what you want to do here is if there's something here, or I say something that doesn't make sense, you want to make note of that. You should also probably make note of what are the really important things I keep hitting on. Well, when something is like, I don't remember that at all, write that down. And then go back later and make sure that's, I don't know is fine for an answer if it's temporary. Between now and Wednesday, we try to take care of the I don't knows. What is biology? Make sure we understand what biology is. We talked a little bit about some of the various subdisciplines. And again, we spent some time between here and the lab. We've tried to introduce to you some basics of the scientific method. Simple process for acquiring new knowledge, testing hypotheses and acquiring new knowledge through that process. And then we working our way up so we stand a chance of actually understanding what the bases of genetics and the bases of evolution are. We have to work our way through and understanding how cells are built and structured. We have to work our way through some fundamentals of biochemistry. To get there, we got to talk about basics of chemistry. So we talked about basics of chemistry, electrons and bonding, atoms, molecules. We talked about polar molecules. We talked about the fact that water is this polar molecule, and that what that means is you have this large oxygen. The nucleus of the oxygen is more likely to attract the electrons to it. And so water being two hydrogens bound to an oxygen, and again, atoms bond through electrons. And because that oxygen is more likely to, those electrons are more likely to gravitate towards the oxygen in this shared relationship, that means that the hydrogen side tends to have a more positive charge, and the oxygen side has a more negative charge. And that allows for what we call hydrogen bonding, where these slightly positive hydrogens and these types of molecules are more likely to latch on to a shared electron in some other molecule. And that's pervasive through all kinds of larger molecules, nucleic acids, proteins, etc. So once we've got kind of a basic understanding of chemistry, very rapid crash course, and we start talking about what are the larger molecules that we need to understand for later on in biology. And there's four main categories of those. You've got your carbohydrates, your lipids, your proteins, and your nucleic acids. We had a quick discussion about monomers versus polymers. Each one of these, what I've described here, basically, they have a monomer unit and they have a polymer unit. So make sure we know what those are. I'm not too picky about what the bonds between them are. The peptide bond is what joins amino acids and proteins. That one's a pretty common thing that you need to know. What's the bond that holds carbohydrates together? I'm not, for me, that's not quite as critical. But knowing what the monomer and polymer units, the common polymers of some of these macromolecules are, and knowing what the monomers that make them up are, that is important to know. Okay. Test one, a lot of topics there. And we got a lot done in a very short period of time. I appreciate you guys suffering through that. As part of that, we had to do online. Cells and organelles. Right, so we talked a little bit about cellular structure, different organelles within cells. What are some organelles that are common within all cells, both prokaryotes and eukaryotes? We talked about, not necessarily organelles, structures. Because prokaryotes don't have organelles. There are some common structures between prokaryotes and eukaryotes. We talked about some organelles that are present in plant cells. Chloroplasts. Most plant cells have got chloroplasts in them. All plant cells are going to have a cell wall. We don't normally see that in any animal cells. There's some other organelles as well that are different. And then chapter 5, the last chapter within that test 1, we spent a little time really focused on the plasma membrane and how the plasma membrane is structured, and so the importance of the phospholipid. For most lipids are non-polar. Right? The phospholipid, on the other hand, has a polar end. That phosphate group attached to that phospholipid has a significant charge that can attract and repel one side or the other of the water. And so phospholipids as a group will interact with water, and what that allows us to do is it allows us then to generate this barrier between the inside and the outside of the cells using 2 layers of phospholipids facing each other. And that allows the cells to control what does and does not enter the cells. Okay. And then we move into test 2. Right, test 2 is really focused on metabolism as a whole. Okay. Test 1 is kind of this introduction. What is life? What are cells? And how are cells structured? And what's going on with the chemistry? Test 2 is the metabolism. What's going on with the reactions within cells? What are the major reactions occurring within cells? Right? So some reactions are what we call catabolic. Some reactions are anabolic. That's the difference between, are we building up compounds or are we destroying compounds? ATP being the major energy transfer molecule throughout the cell. So we talked about that. I think that's, if memory serves, that's where my cat decided we needed some help learning about ATP. So what is ATP? Make sure we're comfortable with that. Make sure we know a little bit about enzymes. Enzymes being a type of protein. We see enzymes all throughout all these processes. I'm not asking you to memorize very many enzymes. And I'm not asking you to memorize any enzymes within the respiration or photosynthesis processes. That's just not, it's not something, well, there is one enzyme, ATP synthase. Take that back. There is one enzyme, ATP synthase, that plays a kind of a linchpin role in both photosynthesis and respiration, aerobic respiration. I didn't put it on there. I need to add aerobic versus aerobic. I'll write that right now. No difference there. Aerobic versus anaerobic respiration. That's rather important. That's kind of covered there with fermentation. Fermentation is where we can repeat the glycolysis process by using something other than oxygen. But if we have oxygen present, a lot of organisms have built up the citric acid cycle. They can use that. That can occur within a mitochondria. Some prokaryotes can generate that process internally without mitochondria. But for us, that citric acid cycle is going to occur in a mitochondria. Fermentation is, and so the citric acid cycle requires an oxygen consumption. Or fermentation can occur in organisms where there's not that oxygen present, or it can occur in our cells too if our cells run out of oxygen, but they need to continue to generate energy. So temporarily they can go through a fermentation process that has to be cleaned up later. And then make sure you've got some comfort with the photosynthesis process. We break that up into the light-dependent reactions and the light-independent reactions. Often we'll refer to the light-independent reactions as the Calvin cycle. But be aware of the ATP synthase and the role that it plays in both, and what powers the ATP synthase. That's just a spinning turbine enzyme that helps us generate ATP using some potential energy the cells have stored up. Okay. It's the hydrogen gradient, by the way. Okay. Exam 3, we're focusing on two main topics. Just a little bit about how cells communicate with each other, and then through that communication process, they can also trigger each other to reproduce, right, through mitosis and or meiosis. Meiosis is pretty specialized. Almost all cells are going to go through mitosis, but again, the communication has to occur a lot of times, triggering that response. Communication occurs at the molecule we call ligands. Right, so ligands. Ligands will bind to either extracellular receptors on the surface of the cell, or in the case of a ligand that cannot pass through the cell membrane, that would need to bond to an extracellular receptor. Some ligands can pass through the membrane, and so an intracellular receptor can receive those signals. Signaling cascade is what occurs after that signal has been received by the cell. So it triggers all kinds of responses. One of those being, probably, I don't know, I may not ask this, but talked about apoptosis. Things I'm noticing I forgot to put on the second. Apoptosis. And again, meiosis and mitosis. We actually saw under microscope. Tried to identify different phases of mitosis. It occurs pretty rapidly. So that's the reason we can't just pick out a single cell and trap it on a slide and say, this one's in anaphase and this one's in prophase. It's doable, but it's not easy. It's a lot easier to just give us slices of cells of tissue that we know is growing. So we know those cells will be going through these phases, and then just go through and try to identify those phases as they occur. That's normally how we'll do that. That's what we did in lab. So be familiar with the phases of mitosis. If you're familiar with the phases of mitosis, it's a little easier to then understand the phases of meiosis because they go through very similar steps. There are some differences. And again, what I would suggest to you, probably the most critical thing we saw later was making sure that the chromosomes and the chromatids, which chromatids are paired, replicated chromosomes. Each side of that pair is a chromatid. Again, seeing what happens and making sure that the exact copies of the DNA get sorted out to each daughter cell through mitosis. Right? Mitosis generates genetically identical daughter cells. It's important we get all the DNA replicated, sorted, and completely equally separated. And then through meiosis, that's the process through which we generate gametes, which are going to be haploid cells. They're going to have half of the chromosomes or half of the genetic material. So we can send that half of genetic material to, or combine it with half of the genetic material from another donor, and that generates a diploid cell. So two haploids together will generate a diploid if we're talking about two gametes forming a zygote. Process of meiosis is similar, and we go through similar phases that we see in mitosis. There are some differences. It goes through two stages to go through meiosis. We talked a little bit about that. So make sure, if I say something that's like, yeah, I don't really remember that exactly, write that down, go back and look at it. That's kind of the goal of what we're doing here today. There's too much material to talk in depth about everything. I'll try to point to some things that I know I would expect to be important. Okay? All right. Test 4. What was the hardest part about test 4? There were several, right? Just looking at your scores, there were several. What was the hardest part for test 4?
Was there blood typing on test 4?
There was blood typing on test 4. Yes, there was blood typing on test 4. Okay. Yeah. What else? Okay, perfect. So, yes, let's keep. So I'm hearing blood types. And Paige, could you hit those top 2 buttons on that panel for me? I would very much appreciate it, ma'am. Thank you. I sincerely appreciate it. So I heard a couple things there. I heard blood types. Focus. Okay. All right. I'm going to switch. All right. So I'm going to switch microphones here. So the sound's going to be a little different, but since I'm going to be over this document camera, I want you guys to hear me. Normally I use the microphone here because it's clearer, but I don't want to be talking right into it and talk loud enough. You guys can hear me, so I'm not talking to this thing now, okay? So whoever's watching the video, hopefully I'm not blasting them anymore. All right, so we talked about blood types, and we talked about I said sex-linked traits. I will not put a pedigree analysis question on the final. Don't discount that, right? You'll need that later, but I don't mind saying I'm not going to put a pedigree analysis on the final. I mean, it's technically a final, test 5. But I don't mind talking about these because these are fair game. I might put something on there about these. Okay. Anything else? Test 4 is sticking out like a sore thumb. These are typical stumbling blocks. I expected, you know, these are not simple concepts. So let's say that I've got what's the best way to do this? You have your genotype and you have your phenotype. Okay, and so the phenotype. So if I have if the individuals are homozygous for AA, and this is in humans, animals, depending on the species, are different, and they have their phenotype is they're just type A blood, right? So type A blood, and we could talk about universal donors, receivers, who can receive, donate to who, and I just thought there's way too much to talk about. That's another time. You're welcome to look that up on your own time. Okay, we've got enough other stuff. I didn't want to confuse the issue any further. But certain blood types can donate to other certain blood types, and some blood types can donate to all blood types, and some blood types can receive from any blood type. And again, if you want to know more than that, I'll let you come stop by and we'll talk more. But it's a little more in depth than we need to be here.
Didn't we have to do that on test 4?
Not about donations. Not about donations. I didn't ask about donating or receiving blood, but we did have to do. I did have a blood type question on there, just kind of checking on dominance and codominance.
So you want to match up like O can only donate?
Yeah, I'm not going to ask that. Okay, I don't mind sharing that. That's good stuff to know, but we have plenty of other stuff to be concerned about. Yeah. That's not to discount it. That's important information. It's not in my outcomes for this course, and so I didn't interpret it as something we have to do. Okay. So genotypically, we can have a phenotype of A in 2 main conditions. That could be AA and that can be AO. Right? So if you get an A allele from one parent and an O allele from another parent, you're still considered type A, and that controls who you can donate to and who you can receive from. Okay. Type B can be homozygous for B. Right? So B and A are dominant over O. So both B and A are dominant over O. So if you have one O allele, that's okay. But if you've got one of these other 2 types, that other one will be your type. That is your blood type. Positive and negative, that's another story. Let's not discuss that. Okay, let's just focus on AO, B, and AB. Now, if you're type AB, there's only one combination that causes that. You have an A allele from one parent and a B allele from the other. That's the only way that works. Okay. There's certain portions of the human population that the B allele is more common in. There's certain portions of the population that the A allele is more common in, and I'm not gonna. We can go look that up, but that's not something I know off the top of my head. I just know that's a thing. Okay. So, questions here so far? So how do we determine, right? So let's say I've got one parent that is type B, but they have an O allele. Let's say I've got one parent that is type A, but they've got an O allele. So they're both heterozygous, but they're for different blood types. Back on the screen. There we go. So how does that happen? So what are the possible combinations for their children? Well, this is the only combination where you could have all 4 blood types could result from this pairing. Okay, you could have, so this individual here could get an A from one parent, a B from the other. Okay, so 25% of their children could be potentially type AB. Here you would get an O, but you would also get a B from this parent, so that would be type B. Right? So I've got a B and an O, so that's type B. Over here I've got an A and an O. So again, the A is dominant over O and the B is dominant over O. So we've got AO, this is type A here. And then finally, there's the possibility of 25% chance, just like the others. This is the pairing that will result in possibly all 4 blood types. Both individuals, both parents are homozygous, and they're different blood types. You can see all 4 of those. Okay. Does this help a little bit? Make some sense. Okay. And again, just drawing your Punnett square, where you take each of the alleles, but then because it's a codominant scenario, it's dependent on the presence of an A or B, right? And I forgot to put on here type O. The only way you're going to get a phenotype of O is if you get an O allele from both parents. So if you have 2 parents that are both type O, all of their children will be type O. If you have 2 parents that are type A, well, it depends. Do both parents carry an O allele? And a pedigree analysis a lot of times is the only way to know that, because at least in modern times. Now, previously, humans would, especially here in this country, we would have, my mom's mother, I think, had 9 brothers and sisters. So when you've got, yeah, we can say there's 25% chance of this particular combination of offspring, and if you only got 2 kids, well, you're not going to know. You can't say with any certainty. You would need multiple offspring to say with certainty whether both parents have an O allele or not. Okay. All right. Questions there? Let's help a little bit. Okay. Sex-linked traits. Okay. So specifically for color blindness, let's say, or we could talk about. Actually, we could talk about calico colors in cats, because I've asked you on the homework the question about the color blindness that we asked on the test. All right, so I'll let you guys kind of wrestle through that. But let's say with cats, let's say that there's black and orange. So let's say B is black, O is orange. Okay. So say I've got a cat that has both black and orange on their X chromosome. It's a female cat has black and orange on their X chromosome. And we've got a male over here that has black on his X chromosome, but then he's got a Y chromosome. The Y chromosome is not going to, most Y chromosomes do not carry, they carry very little genetic information. Can't say none, they do carry some genetic information, but in most species, the number of alleles on the Y chromosome are pretty negligible. Okay? There's enough information on the Y chromosome that it will pair up with the X chromosome during meiosis. There's some crossover, but not much, because there's just not very many alleles. There's not very many genes there. Okay, so what happens then is if you have this female cat, we can put the female across the top row here. We'll put the male cat on along the side column here. So what happens then, so 50% of your offspring are going to be male, because 50% of them are going to get this Y chromosome. Right? And so of your males, if the female is calico, then the males can both be at this, oh, sorry. The males can be black or orange, one of the two. Now, white is another gene on another chromosome. The presence of white fur is another gene on another chromosome that will override in certain cells these other traits. But the black or orange in cats is on the X chromosome. And so a male cat will either be black or orange if the female is calico color. And then 25% of her female offspring will be black. They might be black with white, but they'll be black. And then 25% of her offspring, reasonably rare, will be this female calico color. Again, we're neglecting the white trait. It is a bit of an oversimplification, but as far as the black and the orange, and this works the same way with color blindness in humans, where now this is a shared dominance where black and orange are kind of exhibit this shared dominance, where with color blindness, you've got color blindness as dominant, and the absence there is recessive. Okay. All right. No, actually, it's recessive. I'm sorry. I misspoke. Color blindness being recessive in humans. Questions? Help a little bit. Point you in the right direction. I know now, next time, if I teach this course again in the future, I know now I will spend a good amount of class time really drawing some Punnett squares. I get that. Okay. Okay. Awesome. So let's see what else we got here. Minimize. Let's switch the mics back. So that should put me back on the document camera. Hopefully that all worked. No way to test that until I look at the video later. What did Mendel contribute? Look at Mendel's laws, especially the ones that prevail, the independent assortments. Okay, and again, keep in mind, Mendel did not understand chromosomes were how this genetic information got communicated from one generation to the next. He just was making observations of what he saw in pea plants for 7 different traits. Understand the difference between genes, traits, alleles. We've covered a little bit about homozygous and heterozygous. Homozygous is where you've got 2 alleles the same. Heterozygous meaning you've got one of different kinds. And the phenotype refers to what's observed, what's measurable, where the genotype is actually referring to the alleles that are present. And again, we try to discuss, Mendel's observations didn't cover everything. He was looking at traits that were not linked. He was looking at traits that were very clearly dominant and recessive. He was not looking at codominance. He was not looking at sexual linked traits. He was not looking at kind of the more, I should say, the more common scenarios, because a scenario where I just simply have dominant and recessive is actually not as common. Most of the time it is messy, but the genetics associated, drawing a Punnett square and determining the contributions, how those traits, the alleles are inherited, that works the same no matter how those traits get described in the phenotype. The way they're inherited is the same, whether they're simple dominant recessive or whether they're codominant or they're sexually linked. The way they're inherited is the same. Okay. And what else we have on that test there? So quite a bit on test 4, right? Again, chapter 12 really focuses on Mendel, his research, dominant and recessive traits, understanding how to, and again, I'm not going to test you on using laws of probability, but being able to draw a Punnett square and applying the laws of probability, that's a very simple tool for monohybrid and dihybrid crosses. Monohybrid being a 4x4 square that we've just drawn, dihybrid being a 16 square crossing two traits, right, that we've seen in class a few times. Okay. Make sure we're aware, we spent some time talking about DNA because that's how these traits get inherited. It's passed through DNA of each parent, right? That DNA contains the genetic code. And then the types of RNA we talked about, specifically mRNA and tRNA. Those are the two types that we really want to focus on. Oh, be aware that there's a ribosome RNA and there's a microRNA, but really these two types, messenger and transfer RNA, are going to be our focuses here. Okay. Know a little bit about the structure of DNA, the structure of RNA. Understand whether it's replication, whether it's transcription, or whether it's translation. In all cases, those enzymes, structures, the cellular components will go one direction down those nucleic acids. Normally it's from 5' to 3'. I've used the example before. It's kind of like a cable tie where once you get it started, it goes one way. It doesn't go the other way unless you force it. So understanding the difference between replication, transcription, and translation, that's been present since test 1 of memory serves. Right? That is one of the biggest things we want to come away from this course with. Okay. Let's see what else. Is my keyboard not working? I guess my keyboard's not working. All right, and that brings us then to the new test 5 material, which we ran through rather quickly on Monday. Don't mind kind of going back through some highlights here. I thought Okapi versus Giraffe was a really nice example of two species that are very closely related. A lot of similarities, but a lot of differences. That's because we see speciation here where they diverge, they've gone different directions, but they share a common ancestor, and we can actually measure that today using their genomes. Okay? But we can see that just looking at their traits, dark colored tongues, almost identical skull structures, even though what actually gets expressed externally, their phenotypes are obviously very different. But they do share, we've been able to provide supporting evidence that they share a common ancestor. I tended to cut this slide out. Yes, so not that this is not important, but we've already talked about this. I tended to cut that one out. I thought this is kind of a nice summary. Right? And again, understand that our understanding of evolution started separately than our people that were looking at genetics and the fact that the two are actually tied together. That was not made until later on in science. And all of this has occurred in less than 2 centuries. Well, that's not, evolution technically started kind of early 1800s, so we're right around 2 centuries for both of these. Okay, so being able to come to where we are now, to where we can actually use the genetic code of a virus against it, as we've done with the coronavirus now, we've done that in 2 centuries, which is crazy to me, but that's pretty cool when you think about it. Okay. So again, roughly the timeline. Where does this come in? Now we can actually show genetic codes and actually begin to compare genetic codes for organisms that we assumed had evolved from a common ancestor. We can actually trace that now and show that reasonably clear. Yeah, that keyboard's just not working. Okay. And so these are some of the major terms we talked about last time. Still relevant, still important. Understanding what a species is, hybrid being a cross between species. Natural selection is not evolution, but it is a tool. It is a mechanism of evolution. And there are various mechanisms that we observe that drive natural selection. So again, adaptation is any kind of trait that gets displayed and can be inherited, can be passed on through the material we talked about in test 4, the genetics. So an adaptation is a trait that can provide an advantage to an organism through acquisition of food or its ability to reproduce and contribute its genetics to the next generation. And that's really kind of the driver of natural selection. Natural selection, what we find is that if a particular adaptation is unfavorable in a generation, then it's normally, it gets selected against. Right? It can get selected out. That particular individual, that particular allele may get removed from the gene pool through natural selection. Okay? Those adaptations occur a lot of times through mutation. That doesn't mean mutation is natural selection and evolution do not drive mutation. Mutation is a random event that occurs. It's not necessarily drive that. At least to the best of our knowledge, we've not seen that. But it's this random event that occurs, but its occurrence allows adaptations to surface and then sometimes be allowed to propagate to the next generation. Again, if it presents an advantage, or if it really doesn't present an advantage and it's recessive, then it can get passed on to the next generation, and maybe as the environment changes, then it does present an advantage. Okay. We talked a little bit about divergent versus convergent. So convergent being where we talked about flight, we talked about different species have figured out how to fly, even though they came from different ancestors. That's convergent. Whereas divergent is more like the okapi and the giraffe going in different directions. Okay? Again, evolution is not natural selection, and natural selection is not evolution. Natural selection is a mechanism of evolution. But there are other mechanisms that can cause evolution. Some of those are random, some of those are just happen by chance. But again, natural selection occurs when a particular adaptation is advantageous. Evolution can occur or occurs whenever a particular trait or adaptation is removed or doesn't make it through the generations. So natural selection is a tool or a mechanism through which that can occur, but it's not the only one. We talked about genetic drift. We talked about random events. We talked about bottleneck events. So there are other mechanisms through which evolution can occur. Okay. All right. And again, this video is in your text. If you're following along with your text, you have found it. Maybe you just set aside and didn't watch it. This one's pretty cool. We don't have to watch it here, but I'm serious. Go watch this one. It's like really well done. Is that not gonna open? Anyway, questions for me? If you would please do your course evaluations. If you haven't already, again, I did put out. We had 20 points of homework left to do. I put that out. There's 2 assignments. There's one for chapters 18 and 19, and then there's